So today, hopefully, I'm going to explain the electron transport chain to my mom. So say hi, Mom.
Hi, everybody.
But before we do that, I want to introduce you to what is a cell quickly. I'm not sure if you already have an idea about what a cell is.
I think of the cells sort of as the building blocks of the body, kind of
Yeah.
in animals and stuff.
Yes, totally. So one of the key, I would say the most integral feature of what makes a cell is the fact that it's like a little compartment with fluid inside of it. So think of like an egg, you know, how an egg has like membranes that separate different compartments. A cell is exactly like that. So it has a membrane on the outside, we can see here, and then like in animal cells, for example, there's more membranes on the inside with smaller compartments. So today we're going to focus on a specific compartment of the cell called the mitochondria. And that's this little blue thing. So what is a mitochondria? Essentially, it's another egg. So it has two membranes. So it has an outside membrane and an inside membrane. So the inside compartment is like the yolk of the egg. It's the most inner compartment, and that has a name. It's called the matrix. Then there's an outside compartment, which is the white of the egg, and that very creatively is named the intermembrane space because it's a space in between two membranes. We have two pockets, and they don't mix, except that sometimes there's certain molecules that can kind of sneak through the membrane, and there's also little doors that are kind of stuck into the membrane that allow stuff to move back and forth. So think about a membrane as like a wall with tolls in it. So a lot of things can't move directly through the wall unless there's a toll, but like if you were a bird, you could just kind of fly over the wall and ignore it. So it's like semi-porous, but not to everything. It depends what kind of a molecule you are. Okay, cool. So why does this matter? What does this allow us to do? So think about if you were inside of a crowded train, right? And that train then pulled onto an empty platform. Where would you want to go?
To the door.
Right, you'd want to go out of the train. And it turns out that molecules are exactly the same way. And if they're packed into a small space, they want to move out of that space, and that process is called diffusion. So if we had a bunch of molecules in blue and they're all packed into a space and they can't move between these two spaces, well, then they're kind of stuck and they won't really be able to go anywhere. But if we add some protein doors for them to move through, then they'll move through them, and eventually they'll have equal concentrations on both sides. So they'll be spread out evenly between the two spaces.
So they like to be spread out as evenly as possible, as long as it's allowed with the doors?
Exactly. Going back to the mitochondria, if we do a little zoom in here, we still have our same compartments. So we have our matrix, the yolk of our egg. We have our intermembrane space, the white of the egg, and then we have the outside of the mitochondria, which is actually just the inside of the cell, but we're just going to think of it as the outside for now. And it turns out that mitochondria, instead of having little blue things floating around in it, actually have protons floating around inside. So what's special about protons is they can't sneak through the membrane. They're charged, so they're not allowed to go through the membrane on their own. They can only go through the membrane if there's little protein doors, which mitochondria happen to have. Okay, so they can move through these protein doors, and they do, but there's a catch. These aren't just doors. They actually have a special function, and they're called ATP synthase. So the function is actually very similar to that of a water mill. So the same way that when water flows over a mill, it turns a rotor, which then could be used to grind grain or generate power. When protons flow through the mill to get out of that crowded train and into the open platform, it actually turns a rotor in that molecule, and it actually does physically turn just like a mill. And when you turn that rotor, you generate energy in the form of a molecule called ATP. So ATP itself is not just raw energy, but it is a molecule that has a lot of potential energy stored in it. So I kind of think of it as like a bag of flour. So a bag of flour itself isn't just energy, but like you could eat it and get energy out of it. And it also has a lot of potential to become a lot of different things. So, you can make bread with it or whatever. So ATP is like an energy molecule, the energy currency of the cell is what it's often called. So we want to make it so that we can fuel our cells to do whatever jobs they have to do. Okay, so this is great, right? We have our protons, they're flowing across the membrane, they're causing the mill wheels to spin, we're generating lots of energy, the cell is happy. But what happens as those protons flow? They will eventually come to an equal concentration, just like we saw with the diffusion example, and then the mill wheels won't spin anymore. So then we can't generate energy and the cell is going to be sad. So what do we do? The answer is pumps. So it turns out that the membrane not only has these ATP synthase mills, it also has protein pumps stuck in it. And those pumps, in this case, pump protons and they maintain the gradient. Okay, so this is great. This means that we can keep those protons flowing. They'll flow through the mills, they'll get pumped back down, the mill wheels will spin, will generate energy.
The pumps do what exactly? They
pump protons. So they grab protons that are in the matrix and they move them into the intermembrane space across the membrane.
Thank you. Okay.
Yes, totally.
So they push them back, so then they have to be pushed. So then they put through the mill again.
Right.
And you generate more. Okay.
Exactly. Yes.
Got it.
Okay.
And this is how the cells get energy.
This is how the cells make those ATP molecules to then do things with. Yes.
Okay.
Because it's all about transferring energy from one form to another.
Okay. Yep.
Okay. So cool. And we're going to talk about more about energy in a minute. So the next question is, how do pumps get energy? Because just like in the real world, it takes energy to pump stuff. Like if you were operating this water pump, you'd have to grab the handle and pump pretty hard to move that water. And in the same way, inside the cell, the pumps can't just, work by magic. They need some kind of fuel. So the short answer of where they get that energy from is sugar, specifically glucose. So a very, very simplified version of how glucose gives energy to the cell is you eat it, it goes into your stomach, it goes into your veins from your stomach, and then it goes into your cells. Once it's inside your cells, then it can do chemistry. So when the glucose is inside your cells, it can get broken down by the processes of glycolysis and the citric acid cycle, which I'm not going to talk about, but basically they release a lot of energy. In fact, the same amount of energy that would be released if you just took glucose and lit it on fire. But the difference is, and the reason that the cell doesn't light on fire, is that we do it in very small incremental steps. And what's great about doing it this way is that you can harness that energy to make things like ATP and other things like NADH. Okay, what the hell is NADH? So it's something called a reduced electron carrier, but for our purposes, we are going to think of it as a cow. And there's actually two forms of it. There's the NADH form, which I think of as the sunny cow. It's full of energy. It's like a cow in the morning that's ready to be milked. It has a lot of energy to give. And there's the NAD plus form, which is the sleepy cow. It doesn't have any energy to give. It just wants to go to bed and be left alone. And we can transfer and convert between these two forms.
One has energy and one doesn't, or they both have energy?
So the NADH form has a lot of energy to give, so it can give energy to other molecules, and the NAD plus form doesn't have energy to give.
Got it.
Cool. Okay, so we can move between these two forms, and how do we do this? So we start with NAD plus, our sleepy cow, and we think of those little incremental steps that we use to break down glucose. So it turns out that NADH can actually feed off of some of those steps and take some of that energy and become NADH. And I'm not going to talk about exactly how it does this. It's called redox chemistry, but basically it just goes from the unenergized form to the energized form. So I think about it like NAD plus, there's no milk, it feeds off of the process, and then it makes milk, it's full of energy. Okay, but chemically, what is the difference between these two? And the answer is electrons. So NAD plus doesn't have any electrons to give. It doesn't have any energy to give. NADH does have electrons to give. It does have energy to give. So I'm using milk sort of as an example, but with what it's actually able to give to another process is electrons. And those electrons carry with them energy. So it's a little bit of a sleight of hand there, but I hope that makes sense.
Yep.
Okay. And then you can actually convert back from NADH to NAD plus by essentially milking the cow and giving those electrons to another process. And then this can kind of recycle. So you actually recycle these cows over and over again so that the cell can make energy, essentially. Cool.
Great.
Okay, so how does this let us pump protons? And the answer is the electron transport chain, the title of the presentation. We're finally getting back to it. Okay, so this is a diagram that you would typically see in like a biochemistry textbook, and I'm going to walk you through how we've actually seen everything that's in this picture already. So this is, again, a slice of the mitochondria like the one that we had before. We still have our two membranes exactly as they were before. We have our matrix, the yolk of our egg. We have our intermembrane space, the white of the egg, and we have the outside of the mitochondria.
Got it.
Okay, cool. So we have our protein pumps, which we talked about before, and there's actually a couple of them, and they have different names and slightly different jobs, but we're just going to think of them all as kind of the same. And we have our mill, our ATP synthase, that the protons can flow back through and spin the mill wheel and generate energy.
Yep.
So they have these names. They're called complex 1 through 5 very creatively. And 5 is secretly ATP synthase, but we're not going to worry about that. Okay, so bringing it all together, how do we make ATP? So our sunny cow arrives having fed energy off of the breakdown of glucose. It's full of electrons, full of milk to give to the electron transport chain. So the first pump milks the cow, takes the electrons away. That cow is now NAD+. It's the sleepy cow, so it can go away and get more energy from breaking down glucose, so that's what it's going to do. Okay, so we've now given our high energy electrons to the first pump. What that pump is going to do is it's going to use some of that energy and pump a proton. And I want to make it clear that the pump is not actually using up the electrons themselves to do this. It's just using some of the energy from the electrons to do this. So the electrons themselves don't get used up, but they have a lot of energy, and that energy is getting used up. You pump a proton, and then what happens is you pass those electrons to the next pump. That pump uses a little bit of the electron's energy to pump a proton, and it passes the electrons to the next pump. And this is why it's called a chain, because you are taking the electrons and passing them down. It's kind of like an assembly line. So now we're at the last pump. The last pump is going to use up the last of the electron's energy to pump a proton, but now we have a problem. So the electrons are kind of just stuck at this last pump, and just like an assembly line, if they were to stay at that last pump, we still have electrons coming in because there's more of those sunny cows, there's more of those NADHs arriving and delivering electrons. So if we didn't have a way to get rid of them, we would get all backed up and it wouldn't be able to keep working. So the way that this is solved is that oxygen, which is a molecule that really, really likes electrons, grabs the electrons from the last pump and becomes water. And one of the things that's cool to note about this step, so you know how you and I breathe air? The reason that we have to breathe air is so that we can do this step in the electron transport chain. This is why cells need oxygen, is because of this. So I don't know, I think that's pretty cool. I like thinking about that.
That's very cool.
But
and does the water come out as waste?
Yes, it does. So that's actually right here. So water, as I said before, can actually kind of sneak through the membrane. So it actually does that. It sneaks through and you actually breathe it out in your lungs. Okay, so now we have generated our proton gradient. We have all of our protons packed into the intermembrane space, a low concentration of protons in the matrix. If we remember our packed bus example, they're going to want to get out of the bus. So the way that they do this is they go down through our mill, through ATP synthase. The proton moves, it turns the mill wheel. When the mill wheel turns, That allows us to make ATP, the energy molecule of the cell. And because we have lots of protons being pumped into the intermembrane space, lots of them can flow down through the mill, and we're going to generate lots of ATP. So now we have a ton of ATP in the mitochondria. That mitochondria can then deliver that ATP all over the cell. Our cell has lots of ATP, it has lots of energy, it's happy, and that is the electron transport chain. So now I have some questions. Hopefully they should be not too bad. Okay, so the first question is about these guys. So it's, what is the purpose of the cows and where do they get their energy from?
The NAD plus cow, and I hope I'm saying that right, gets its energy from the glucose, which is
Yes.
From sugars that we eat, I think.
Totally, yes, it's totally, yes, from glucose.
And then it sucks them up, to use a very biological term, and turns into NADH.
That's right, yes.
And then it has the energy. Sorry, and what was the other part of the question?
What is the purpose? So what do they then go and do with that energy?
So then the cows, the NADH, which has electrons to give, goes to the pump and shares its electrons.
Yes, totally.
It's an electron carrier. Yeah.
Yes, yes, awesome. Oh my God, this is awesome. Okay, so next question. You kind of talked about it already, but so where do the pumps get energy from to pump protons?
From the NHD cow that it has electrons to give.
Yes, totally, from the cow. All right. Why does it matter that we have a proton gradient in the mitochondrium?
It sounds like it is, it seems like it's part of an energy producing system, and to keep equilibrium in that system, from what you've told me, that it kind of moves the energy back and forth in the cell and keeps everything equal, but also generates energy in that process.
Yeah, no, you're definitely onto it. Think about the mills. Does that help?
Well, the mills are to create that equal balance across the.
So yeah, the mills allow the protons to flow, and when the protons
so that they
flow,
can be. It creates the ATP. Is that what it's called?
Yes, yes, totally.
Yes, to feed the cell.